This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 69, recorded November 21st, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered is Elio Schaechter.
Well, how are you doing? Nice to have you back on our shores. I know you were in Germany for a while.
I was in Germany, and then the week before I was in Madison, Wisconsin.
Okay.
Where there are a lot of people who know you, Elio.
Oh, no.
Yeah.
I hope they didn't tell the truth.
You're well known everywhere. In Germany, I was at a meeting on giant DNA viruses.
Sounds scary.
Can you imagine that there is a virus with a 2.5 million base pair genome, and you can see the particles in the light microscope?
I got an email, the blog got a comment from Jean-Marie Claverie, who's one of the people involved in this.
He was at the meeting,
He was at the meeting, and he points out that the lack of knowledge of these things is directly due to the fact that viruses were supposed to be filterable. That's what Ivanovsky discovered in 1897 or so, 1892. And therefore, if something is not filterable, it cannot be a virus. These guys, of course, are much too big to get through a filter.
That's right.
So it's a very nice example of where a dogma is in the way of the truth.
That is absolutely right. And in fact, I did a podcast with Jean-Michel Claverie while I was there. And that'll be out on TWiM. And he talked about that because I asked him, why are we only learning about these? And that was one reason. And the other is that, we've looked at viruses in terms of making disease a lot. So that kind of biases, and environmental virology has not really gotten a lot of attention. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
How you doing?
Great.
How's your handicap?
Golf season is just about come to a close here in Michigan.
Yeah.
I'm sad to say.
And you don't play all winter?
It's totally dependent on the temperature.
Yeah.
So
well, you could,
I'll keep you posted.
If you went somewhere warm, you could bring your clubs, right?
Oh, of course.
Yeah.
Okay. Thanks for asking.
You never actually mentioned your handicap on TWiM. You will not reveal it to us, right? It's like
low 20s.
Low 20s. It's like asking your age.
Yeah.
That's what you said once.
That's right.
Also joining us today from Yale University is Joe Handelsman. Welcome back, Joe.
Thank you. Hi, everybody.
So nice to have you back, Joe.
I know you're very busy and therefore have not been able to join us, but whenever you can, it's great.
Well, thanks. Today
we have a couple of papers to do for your listening pleasure, as always. And the first one is in an interesting journal, which I generally don't read, but I do scan the table of contents. It's PLOS Computational Biology. Any of you ever scan this journal?
Do not.
No. Well, this paper is called Bacteria Human Somatic Cell Lateral Gene Transfer is Enriched in Cancer Samples, and that caught my eye. And it is by Riley, Sieber, Robinson, White, Ganesan, Nurbach, and Hotop. And it is an entirely in silico analysis, which I suppose is why it's in PLOS Computational Biology. And I think it really illustrates the power of bioinformatics, as well as some of the limitations. And so what the idea is here is they were looking for the presence of bacterial DNA in human DNA. As you know, we have lots and lots of data from sequencing human genomes, and they asked whether you could get lateral gene transfer of bacterial DNA into human DNA. And we know that under certain conditions, this can happen. And it's an interesting question because, as everyone knows, we have lots of bacteria in and on us. Our microbiome Is that the right word, Elio? Can I call it a microbiome?
Yeah, I just don't know how to define it yet, but we're working on it.
We know. It's just other people don't.
Apparently.
I see.
And so the likelihood is that our cells take up some of this DNA, and whether it gets integrated or not into us is a good question.
Actually, if I may, they make I think the very good point that proximity counts. And that the transfer is more likely to happen when when our cells are next to bacteria, and of course that's the case in our body.
Right.
So it's not just a DNA floating around in the ocean and we drink it occasionally or something like that. It's that we are very near the source of the genetic information.
Yeah, that's right. So we have plenty of microbes right next to our cells, all throughout our body. So can that DNA get in there? And they cite a couple of interesting examples of known cases where bacterial DNA can get into other cells. And I guess a good one is the bacterium Agrobacterium tumefaciens, which puts its DNA into plant cells to induce a tumor, and a couple of other examples as well. And of course, you can put bacterial DNA into eukaryotic cells experimentally as well. So they set out to ask, can we find bacterial DNA in the human genome? And one of their ideas is that maybe if it does integrate, it's mutagenic, all right? Because we know that viruses, for sure, some viruses integrate their genomes into our genomes. Retroviruses, of course, integrate, and the human papilloma viruses, when they're associated with human cancers, there's typically a piece of the papillomavirus genome integrated into our host cell, and that transforms the cells. Hepatitis B viruses and others. So these are examples where the integration of viral DNA into our genome is oncogenic. So the question is, can bacterial DNA have the same effect? So there's a lot of sequence information available online from a number of different projects where the human genome has been sequenced, and they initially look at a couple of them. And this paper is interesting because, as you'll see at the end, a big problem is figuring out whether, if you find bacterial DNA in human genome sequences, whether it's real or it's an artifact, because sequencing is so sensitive, but it's also fraught with various problems that just finding bacterial DNA by itself is not enough to be conclusive. So they developed an algorithm where they could scan sequences and then pull out, say, bacterial sequences, and they validate this program with Wolbachia, which have endosymbionts, and they can find evidence of these endosymbionts in the chromosomal DNA. So they feel that the system is validated. So then they use their programs, and there are lots of.
You want to say the Wolbachia? Excuse me, Wolbachia is the endosymbiont.
Wolbachia is the endosymbiont of insects. Yes, correct.
Yep.
So they can find Wolbachia DNA in the insect hosts, and these sequences are out there, so they can apply their algorithms to that data, and it works. They can see the Wolbachia sequence in the insect genome. So they used, now it's interesting that when you generate sequences of humans, you can either get blood from people, and typically you take the lymphocytes and purified DNA and you sequence that. Often you get sperm. You can get sperm if you want to look for somatic integration. Genomes integrated into germ cells. Or you could have a cell line made from a person. In many of these genome projects where they're sequencing a lot of people's genomes, they make a cell line from you. They take some cells from your cheek and they put them in culture and they immortalize them, and then they can always go back to them. And so every project has a different kind of source of DNA. And as you'll see, that could be a problem, because if you take blood cells from a person and you make DNA, that's it. You can't go back and get more. So the sample is destroyed in the sequencing. Whereas in a cell line, you always can go back and get more of the DNA, but that can be fraught with contamination issues, because as soon as you're growing cells in a laboratory, it could be contaminated with all kinds of other DNA that you have around. All right, so they use their algorithm, and the first set of data they look at are some older sequences of the human genome, which were made by Sanger sequencing. And the reason I say Sanger sequencing is because Sanger just died recently. It was maybe yesterday or the day before. And Frederick Sanger had 2 Nobel Prizes, one for developing protein sequencing and one for developing DNA sequencing. Anyway,
Impressive.
right? And
Not
apparently he was 90-something years old and was still working in the laboratory.
Bless his heart.
Working on his 3rd Nobel Prize. So the early human genome sequences were basically you take human DNA from blood or sperm or cells, whatever, and you clone it into a bacterial plasmid, and then you sequence each plasmid. All right, so this involves cloning. And by looking through those sequences, they found they have 113 million individual Sanger sequence traces. This is from 13 sequencing centers and over 8 individuals. And then they had 2,241 bacterial genomes, and they say, are there any of those bacterial genomes in the human sequences? And they found 680 sequence traces. Again, these are human sequences that contain bacterial sequences in them. And most of them were species of lactobacillus. Okay. Now, they also found E. coli and Erythrobacter, but they throw those out because they feel that's contamination. E. coli, of course, because everybody has E. coli in their lab, and if you're cloning the human DNA for this sequencing project, it's likely that you got E. coli DNA in there as well. And they also feel that the Erythrobacter is contamination as well. So that leaves us with lactobacillus, which I suppose makes sense, right? Because we have lactobacilli in I, right?
Yeah, we do. And I think almost all mammals have some sort of lactobacillus species.
Right. So that's the Sanger sequence, the cloning. So they feel that this is suggestive, but because there are so many problems with sequencing clone material, they're going to kind of put that aside. And so then they go on to the next sequence project, which is called the 1,000 Genomes Project. There is an ongoing project to sequence 1,000 human genomes, and this uses next-generation sequencing. And here you don't have to clone the human DNA. You fragment it, you add linkers to the ends of the DNA, and then it can be read, it can be sequenced in a machine. And the sequencing used for the 1,000 Genome Project is called Illumina Sequencing because it uses a machine called an Illumina machine. And here they have lots of sequences, and they calibrate this. They have a software algorithm again for comparing the human sequences with the bacterial sequences, and they calibrate it using HeLa cells. The HeLa genome has been sequenced, and we know that integrated into the human genome, among other things, is human papillomavirus. And of course, everyone must know the story that HeLa cells were made from Henrietta Lacks in the 1950s. And Henrietta had a cervical tumor from which she died, but some cells were taken from the tumor, and those were the source of HeLa cells. And we now know that she had a papillomavirus DNA insertion into her tumor. So they could use their software algorithm to validate the presence of human papillomaviruses into her genome. And it's known that it's in chromosome 8, and they could see this by their sequence. So their programs work. So now they apply it to the human sequence. They have 3.15 billion Illumina read pairs. Now, in the Illumina sequencing, what you do is you take human DNA, you break it up into smaller pieces, and then you add linkers, which you then attach to a solid surface, and then you do the reactions. And you can do it in a couple of ways. You could read just one end of the DNA, or you could read both. And when you read both, it's called a paired read. So they have over 3 billion read pairs from this 1,000 genome projects. And of those, 3.15 billion, their software showed them that 7,191 showed evidence for bacterial integration. Okay, so just scanning all the reads for bacterial sequences in that Illumina database of over 3 billion reads. They find over 7,000 which have bacterial DNA in them.
Which makes it a very rare occurrence.
Yeah, I would think so, yeah. And the most common is our species of Bradyrhizobium. And I think, Joe, you would like that, right?
Yeah, I found that to be one of the most curious aspects of the paper, because Bradyrhizobium is in the same group of alpha proteobacteria as the Agrobacterium, which of course is the classic example, as you mentioned, of DNA transfer from bacteria to a eukaryotic host.
Yeah.
So I wondered if maybe there's something special about this group that they somehow know to attach well to human cells or plant cells or whoever their host might be.
Is this something you'd find in the gut microbiome?
I was wondering the same thing. I have not seen it myself, and I might not have been looking for it because it's certainly not one of the things you would expect in the gut microbiome. First of all, it's aerobic, and a good portion of the gut microbiome is anaerobic, and Bradyrhizobia are really associated with environmental samples and plants. So it might be there, maybe I just never noticed. But
yeah, so
it's an interesting find.
They note that it's unusual, but they note also that most of the samples that were positive for Bradyrhizobium came from a Chinese population. These were samples sequenced by the Beijing Genomics Institute. So they suggest that it has something to do with diet. And maybe their diet contains these bacteria, and that's why this is there. And this is actually an interesting point they make, that when you sequence diverse human samples, you don't always know other things, diet, lifestyle, etc., that's associated with them. And those, of course, could influence the microbiome, right? So,
most assuredly does.
Yeah.
And it might introduce bacteria, but I think it's unlikely that Bradyrhizobia or alpha proteobacteria like that would make it through the stomach acid. Maybe they would, but it's unlikely. But I think more likely is that the change in diet could select for growth of one group of bacteria.
Yeah, right. So they also found 2 other kinds of bacteria. Actually, they called them OTUs. Maybe, Joe, you could explain to our listeners what an OTU is.
OTUs are operational taxonomic units, which sounds very complicated, but it's really not. It's an operational definition of something that we can't define, and that is what a species is or what a genus is in bacteria. We really don't have good descriptions at that level. We don't know at what level of difference we cut off a species or a genus. And so for those organisms where we haven't been able to cultivate them and we don't know more about them than just a sequence, we assign them to OTUs, and to be full, an OTU has to have some sort of number attached to it to say whether all the members are 100% identical or within 97%, which is typically considered equivalent to species, or some other level of similarity.
On the other hand, they give you the names of those 2 OTUs. Propionobacter acnes and Enterobacteriaceae.
Yeah.
So I don't know why they call them OTUs, but it's okay.
They shouldn't. Yeah, they shouldn't need to, except that some of their other sequences were unnamed. So perhaps they were just referring to all of the sequences as OTUs. And oh, by the way, one of them or 2 of them could be taken to real names.
So one of them, Propionibacter acnes, they think that's a contaminant. It's on everyone's skin. And so they think that, whoever is preparing the DNA samples contaminated the samples. Remember, these are just the presence of bacterial DNA sequences in the human sequences. So these are typically short runs, or short reads as they called. So you would just get a bacterial sequence. So
but it does help in the sense that you do want to have some sort of a feeling for how often is contamination to be likely. This tells you that it's likely. Of course.
They also think the Enterobacteriaceae is contamination with E. coli for the reasons we talked about before. So they actually throw out all hits of P. acnes and E. coli or Enterobacteriaceae, even though some of them might be valid. I mean, this is a very prevalent issue in this paper, and they have whole sections on contaminations, laboratory artifacts. In fact, the next section they talk about trying to devise algorithms to see if the hits they're getting, that is the bacterial sequences and the human sequences, if they are artifacts or not. And so they use one way which puts all of the bacterial sequences into one of two classes. One where, so they look at the contribution of bacteria relative to the total population of bacteria, which is higher in reads, or one where it's higher in reads coming from the microbiome. So if you just look at the proportion of bacterial DNA, either with respect to the human genome or with respect to the microbiome, they feel that can give them an idea of some whether it's contamination or not, right? So
you're talking about specific bacteria.
Specific bacteria, right. So they have a very nice graph where they plot these two ratios, the proportion of these OTUs compared to human versus the microbiome. And the Brady rhizobium is much higher in this category A, which is relative to the human sequences. So in category B, which they would consider a contaminant, they have the Propionibacterianae, and Enterobacteriaceae, category B. And another one there is Xanthomonidaceae, which they think is all contaminants. So Brady rhizobia, category A. And a big surprise, phage lambda falls into this A category, which they think is real.
I figure that out. Why would they think the coli is a contaminant, but the phage lambda is real?
Yeah, I mean, strictly based on this ratio, right? Because
statistical argument.
Yeah, it's a statistical argument. Sure, they don't know, and in fact, they end up saying that they seem to think the lambda might be real, and they think it should be validated. Mm-hmm. So that's curious, anyway. Rate of integration, they can, from the numbers, they can look at how often you pick up bacterial DNA. So the trace archive data, which is the Sanger sequencing data, they had 680 integrations in 113 million reads. So that's 6 times 10 to the minus 6 integrations per generation. And the Illumina data, which is the 1,000 Genomes project, it's similar, 2 times 10 to the minus 6 integrations per generation. And that's on a higher data set. It's 3 billion, if you remember. 7,000 out of 3 billion. So they think that that's a good sign that the rate of detecting bacterial DNA and human DNA is similar when you compare these two very different sequencing projects. Another issue that is really interesting is the coverage of the DNA sequence. And the coverage simply means, in its simplest way, how many times you sequence a given sequence, or how many times you determine the sequence of a given region. So not really good coverage is 1x, which means you basically have one sequence run for every area of the genome, but 100x is really good. And it turns out that the 1,000 Genome project isn't very good because it only has about 1x coverage. And having a lot of coverage would in theory allow you to see the integration sites of the bacterial DNA in the human genome, right? Because then you can assemble longer sequences from the short runs, and you can say, here's the beginning of the bacterial sequence, here's where it joins human DNA, and here at the other end. So that would clearly be better than simply having a piece of bacterial DNA in the human sequence. So if you look for in this 1,000 Genome projects, for greater than 1x coverage is pretty rare. So now you only get 275 read pairs that are bacterial DNA. And again, that went down from just over 7,000. So that may be one way to limit contamination. And when you do that, the most predominant species level OTU is Stenotrophomonas maltophilia.
It's good.
Which I had not heard of.
Well, that's a nasty pathogen.
Yeah.
You don't want to associate with that guy.
So they call it an emerging pathogen of the respiratory and blood systems of immunocompromised people.
Huh.
But interestingly is another one that originates often on plants. So it
Really?
seems like plant-associated bacteria keep coming up here.
Interesting. Now, the curious thing here is that so the bacterial reads, the sequences are distributed across the bacterial genome, but most of the reads, that's 100 read pairs, came from 2 people in a single study. So, it could be that one of these people had been infected and picked up the DNA, and that's kind of biasing the whole thing. But obviously, you need to do more of this. We don't have that many genomes. That's the problem so far.
Right.
So they say, given the low coverage, the data from the 1,000 Genomes Project is inconclusive in the absence of experimental validation. Can you imagine? So far, everything we've talked about, they feel is inconclusive. Now, remember, we don't have cell lines for most of this data. We just have blood, and that gets destroyed in the process of sequencing. And if you derive the sequence from cell culture, there's always the issue of contamination. And this is why they moved to the tumor cell genome archive. So this is another set of human sequences taken from human tumors of various sorts. And here we have over 7 trillion bases of Illumina paired end sequence. So not just single sequence runs, but paired ends. And in this case, 691,000 read pairs were in support of bacterial integration. The cool thing about this database is that it has over 100x sequencing coverage. So
100x?
Yeah.
God.
So you can start to get the integration sites. If you can assemble, consecutive reads, you can see, as I said before, the integration site of the bacterial DNA into human DNA. So an interesting number, 64% of these tumor samples. So in this database, this tumor database, they often have tissues from non-tumor tissues from the same people, from which they get sequence, and then they compare it to the tumor. So 64% of the DNA in this database is from tumors, but those DNAs had 99% of the bacterial reads. So there is a bias towards having bacterial DNA and tumor DNAs, just from that number alone. And a couple of interesting observations. The most common cancer that had bacterial, or I should say the most common integration or presence of bacteria, it was in acute myeloid leukemia, 665,000 read pairs, which had bacterial sequences. And in this case, in this tumor, Acinetobacter was the most common integrated or DNA present. And
another environmental.
Yeah. Followed by Moraxellaceae. Does that ring any bells?
Well, Moraxella is an eye pathogen, among other things.
Okay, so this
they
is
call red eye, I think.
Is its natural reservoir soil or?
Gosh, I don't know.
I think Moraxella is more often an animal associate, but that would be worth knowing, just because of the pattern that are to be environmental.
One of the interesting observations within the Acinetobacter genome, the reads were often in the ribosomal RNA operon.
Yeah.
So not the most highly expressed bacterial genes, but rather ribosomal RNA seems to be integrated frequently. Also, the location of integration. Now, remember, because these are high reads, paired end reads of high coverage, you can start to see the integration sites. So
they make a point that ribosomal RNA is not recognized by the immune system as well as some other sequences that are commonly recognized. Yeah. I thought that was interesting. That was a good point.
Yeah, so it could be that RNA persists and therefore can integrate.
That's right.
Yeah.
It's just not noticed and not hit.
Right. And in case anyone was wondering how RNA could get integrated, well, all of our cells have reverse transcriptase in us. We have encoded in our genomes various reverse transcriptases, so they could copy that RNA into DNA and then it could integrate. So that's completely plausible and is thought to account for a variety of, for example, repeated elements in the genome. Location of integration, most frequently in the mitochondrial genome. Not in our nuclear genome, but mostly in mitochondrial genome in the cases where they can identify the integration. The next most common tumor that had bacterial DNA was stomach adenocarcinoma with 11,000 bacterial hits. And that was followed by breast cancer and ovarian cancer. And here in the stomach cancer, the most common bacteria OTU was Pseudomonas, and in particular Pseudomonas fluorescens. And there they could see the ribosomal RNA of 184 ribosomes, which is 98% of all the reads going from Pseudomonas ribosomal RNA.
And again, it's curious. That's thought to be an environmental microbe, not something that colonizes humans all that frequently.
Very common on plant roots.
Right.
So once again, plant-associated bacteria show up. It's very curious.
They make a point of saying that they only found 2 Helicobacter reads, even though Helicobacter is associated with stomach cancer. Doesn't seem to be integrating in the human genome. Now, finally, what genes are the bacterial sequences integrated into? 5 nuclear genes have the highest integration rates, and these are all genes that are known to be upregulated in stomach cancers. They involve a lot of what are called CEA, carcinoembryonic antigens, and a lot of proteins that are involved in adhesion.
Did you think that this was a particularly cogent argument for this not being an artifact? The specific integration into the genes that...
I think so, yeah.
Yeah, I think it's quite amazing.
You would expect it to be random, right? If it
Absolutely,
were, if it were a laboratory artifact, for sure.
Right.
And, so they're very cautious in the end. They are saying, look, here's one of their conclusions. Based on our analysis, it is impossible to determine if lateral gene transfers have a causal role in cancer or are simply a byproduct of carcinogenesis. I think that's probably appropriate caution, right?
I think it is, because it could be either way. You could argue that integration causes cancer, or cancer just carries integration along.
Right.
Or the event that triggered the tumorigenesis, let's say some DNA damage that caused a lot of double-strand breaks, would then create an opportunity for non-homologous DNA to be integrated through non-homologous end joining pathway. So it could be the 2 events were coincident and not causal.
Right. One of the things they point out is that for many of these tumor samples, they don't have a normal tissue control. So they have some, but not all. You would like to have a normal sample from the same person. They don't have it. And they said they tried to do it, but they were forbidden to do that because of IRB rules. So, there are a lot of questions that you can't answer because these are patient samples and they are coded, and you can't pursue it without your own protocol. They spend much of the discussion talking about possible contamination, and they're very worried about that. But they do feel that there's some good evidence that there is some specific integration, and they say it's worth. These data show that it's worth pursuing this issue, whether the integration of bacterial DNA is oncogenic or not. I don't know. What do you think would have to be done next to really address this? I guess you'd have to get people with tumors. You'd have to enroll people with tumors and get normal and tumor tissue and do this sequencing from them at high coverage, right?
And I think if you did it over time and found the same sequences in the same place, it would be really compelling, because I think it's the reproducibility that is the most powerful in the current paper and would also enhance any future analysis.
Right.
I still find it very puzzling that most of the species they identified are not bacteria that we know of to be in high numbers in the human microbiome, which would have been my assumption going in.
Well, maybe you have to ingest naked DNA. Maybe their DNA is in our food, and they get in somehow that way.
Right.
I mean, maybe also a part of a study would be to sequence the microbiome of the individuals whose tumors you're looking at, right? Mm-hmm. Just to see if the bacteria you're detecting are actually there or not.
Right. And again, of course, when you say the microbiome, it would have to be the local microbiome, wherever you collected the biopsy. You'd want to get normal skin, for example, because the microbiome in the gut is going to be different from behind the ear, etc.
Wow. So if you have a stomach cancer, that'll be tough. You have to go in with a sampler, right, to get the microbiome there.
Yeah, but if you're sampling the tumor, then hopefully you could sample healthy tissue nearby.
Right.
Right. So you would actually sample the microbiome from the tumor tissue and the normal tissue, right?
Yep.
That would be a valid way to do it, right? So I guess you could just extract total DNA from the tissues and segregate the sequences according to bacterial or human, right? That would be the way to do it.
Yeah, get the computers humming.
They are.
Do all that computational analysis.
They are humming. I mean, that's the thing here, and this is maybe the last point we should emphasize. We're talking about, trillions of sequences that can only be manipulated by computer, and you have to have a leap of faith to trust that your algorithms are right. You're going to pick up very rare things that are hard to validate experimentally. So this is tough stuff. This is certainly not what I was trained to do. Or probably Elio either, right?
Oh, this is smack in my comfort zone. I read these things and I say, gee, I should start all over.
It also kind of gets at the heart of an issue in science, in that it's harder to prove things that people don't know already or already have some hints, because we're so biased. As a PI, you'd like to see, as proof of principle, something come through the screen that you would have expected based on your prior knowledge.
Yeah.
And if you demand that, then as they point out in their discussion, you're really closing the door to a new area, a new discovery of research if you don't pursue it and really test in a rigorous way.
Well, I think we have to be willing to accept risk when you do these kinds of experiments, and I think it's getting harder to do that. But this emphasizes to me the need for, I would say most graduate students as part of their PhD training should get computational training these days.
Oh, I think they do.
But here in our
program,
we don't in our department.
Oh, they do it when you're not there.
So, Joe, your lab does a lot of this sort of computational work, right, with your sequences?
Yes, absolutely. Yeah, I am one of those poor souls that does read PLOS computational biology on occasion. So, yeah, I'm a little with this, but I can't really do the analyses myself. Mm-hmm. And I'm in the same boat that you are, but my students are extremely well schooled in it. And I've learned the right questions to ask. I just can't answer them. So I get experts to help with that.
So how do your students get to learn? Just by hearing people who know how to do it? Like your collaborators, for example, they collaborate with them and they learn how to do the work?
I try to have a computational expert in the lab at all times. Okay. And I have one right now, Ashley Shade, who for the moment is with me, and then she'll be moving to Michigan State next year, and I will lose her and have to find somebody else to do this. But she offers the most wonderful workshops on computational methods. And has the students, who are often faculty, professors, postdocs, all sorts of people, but the students of the class actually go through analyses for a few days with her. And every one of them I've talked to said they feel really confident coming out that they can do at least a basic computational analysis of sequence.
And to echo that, one of Joe's former trainees, Pat Schloss, is a tenured professor in our department, and he also runs workshops. So he invites workers who want to move into this area to come in and get an intensive workshop so that they can be competent to use some of the tools he's developed.
And it's really important. Well, those PhD students listening, learn this stuff. It's really going to be even more and more important going forward. Okay, I think that it's a really cool paper. It's in open access, so everyone can check it out. You can see how things are going here in this microbiology field. The second paper is in Applied in Environmental Microbiology, which is an ASM journal. And it is, I would say, less computationally intensive, but nonetheless interesting. And Elio, you're going to tell us about this?
Okay, let me read the title first. It's called Specific Midgut Region, Controlling the Symbiont Population in an Insect Microbe-Gut Symbiotic Association. It's by a group of workers whose name I won't read, from South Korea and some from Japan. And they're dealing with an interesting topic, which is symbiosis. This is not endosymbiosis, which is something we've talked about, and which is one of my favorite topics, of course. This is an ectosymbiosis, that is, the symbionts do not crawl inside the cells of their host. They just simply sit on the surface. Now, the story here is that we have a lot to learn. You would think that agriculturally important bugs like the bean bug, Riptortus pedestris, which is an important pest of beans of all sorts, would have been studied into the ground, and it has, but there's still surprises. And one of the surprises is that in the midgut, which is divided up into 4 different regions, one of the regions is a double region. That is, they found a new region. The names they give to these things are M1 through M4 for midgut. And the region that's called M4 has actually an M4B little region in front of it.
And B stands for bulbous.
For what?
Bulbous. Evidently, anatomically, they see a bulb emerging from that region.
Right. Okay, that's the reason. Okay. Yeah. Anyhow, the gut of an insect is not simple. This is at least as complicated as our gut, only on a smaller scale. But there is this region called MB4, and this is what they are studying here. So basically the message is that in region M4, that is the one that we knew about, there's a high population of a beta proteobacterium of the genus Burkholderia. In the very old days, I used to think of that as being a cousin of Pseudomonas, but it's not, far from. Anyhow, they are present in large amounts in region M4. But in region M4B, you find them, but they're not alive. That is, there are bacteria, the same bacteria are there, and they are not able to make colonies. We'll come back to that, won't we, Michele? You want to talk about that. What does that mean? But let's wait for a second. So the idea is that, by the way, M4B comes before M4. Okay, so think of it as something that happens before M4.
Before meaning as food is coming in.
As food is coming in, right.
It goes first
to the mouth.
Yeah.
It's towards the mouth, and M4 is towards the anus. All right. So they now say, how come these bacteria and they're dead? And so they found that, in fact, there is an antimicrobial agent which is not identified, I'll hasten to say. They don't know, they don't call it an antibiotic. It happens to be heat sensitive, so it may not be a small molecule, but they don't know that. Anyhow, they make lysates of these regions, and the lysates from M4B are effective against Burkholderia, and not against E. coli, and not against Burkholderia, which has been grown in the laboratory. So what they're talking about here is extracting the Burkholderia from the M4 region and assaying it essentially directly. Whereas if you go through the laboratory and culture Burkholderia, it's different. I find it very puzzling. I don't think they really know what's going on. And it may have to do with, who knows, maybe what happens before in this M4B region where the same bugs are present but not alive. I don't really know.
So, Elio, this cultured Burkholderia, right? Is this derived from the gut, or is it just a standard?
But now you're asking a good question. I think it's the standard strain.
I see. Okay.
Yes.
Yeah. Did you pick that up any place, Michele or Joe? I'm not sure.
Now.
I thought they said that it was a lab. They called it a laboratory.
Oh yeah,
strain.
okay. So it's an old, yeah.
So it's
right.
it's clearly different from what's in the gut then, yeah.
It's fairly different what's in the gut.
Yeah.
But and they don't really do the experiment of taking the bugs from the gut, growing them for a while in the laboratory, and then assaying. I didn't see that.
Right.
That would be an interesting experiment to do. Anyhow, this is about it. This is about the story, and it's an interesting story because it shows you that the population dynamics, if you wish, of symbionts in insects are far from being simple. So they certainly are eager to identify the molecules involved. They don't think it's a peptide, as I say, because it's heat sensitive, and they're going to do a lot of work. They're not eliminating the possibility that these things are related to lysozymes or cathepsin proteases or such other defense-related proteins.
So why do you have to? Why is it necessary to have some antimicrobial agent to inhibit these bacteria?
Well, this is really a good question. I mean, I was puzzled. I thought that maybe if I were designing this, I may design the 4M4B region to be after and not before the M4 region. I mean, why do you want to get rid of the bugs before the place where the bugs are present and probably do some good?
Yeah.
Oh, and by the way, I didn't say, let me back up a second. It's not entirely clear what all these bugs do, but if you sterilize the insects, presumably by using antibiotics to get rid of the Burkholderia, you find that the growth is retarded, nymphs have a higher mortality, and sterility is affected. So you have these are all arguments for saying that these bugs are beneficial. Yeah. So if they're beneficial, why would you want to sort of kill them before?
Yeah, it's puzzling, isn't it? Because they imply that there's been a long coevolution. So it seems to me the bacteria would have developed resistance. So I'm wondering if what they're really looking at is viable but non-culturable bacteria in the M4B region. So
why don't you enlighten us a little bit about that?
A developmental state you can think of it like a spore-like state, a highly resilient form of cells that don't replicate, but they're very hardy.
And there would be a reservoir. There would be a way to make sure they're there.
For the bacterium, it would be more of a détente, let's say, between the host and the microbe. Would control the numbers, but the bacteria wouldn't die. I think to test that idea, they could do the reverse experiment. So they could harvest the bacteria from M4B, where they get no colonies, but then incubate them with a lysate from the M4 region. Which is a region that is supportive, and ask if they can now resuscitate.
Yeah, that would be good.
Those cells would be one kind of pilot experiment I would try.
Yeah, that's good.
Not bad.
If this is representative of vertebrate strategy to control overgrowth of bacteria that we don't think that much about, because so many of us think in terms of antibodies and the adaptive immune response of mammals, which is so important for keeping bacteria in check. But in insects, they have very few ways of controlling bacteria, and they seem to be somewhat nonspecific in their responses. And so I'm wondering if this organism could just overgrow and become a pathogen, since it is an opportunistic pathogen in other organisms. And this is a way of suppressing the populations, and that, some of it is needed, but any bacterium that grows too much in the wrong places will be pathogenic to the host.
Yeah, so if they can identify the antimicrobial agent, in this case, they could try and remove it and see the effect on the insect.
That's right. I don't know if this insect has a genetic system.
Yeah, it might not.
It may not, but it may, and I just don't know.
But you could remove it. You could remove it with RNA interference. Right.
Of course.
And then see if the insects get sick or something like that. Yeah.
Well, you have to know a little bit more about the nature of the compounds involved, like you say.
Yeah, I mean, it may be a chemical, in which case it's harder. You have to go into the pathway that gives rise to the chemical. But either way, I suspect that's where they're headed, in part.
It might be possible, if it is a small molecule, to select for resistance. And even though it hasn't risen in this environment, the concentration is low enough that the frequency of resistance is also low enough that maybe they're just not seeing the resistant mutants. But that would be a really interesting experiment to replace the normal bacteria with the resistant ones and see if they overgrow.
Yeah, that would be easier than knocking out the antimicrobial itself.
Yeah, they also, I know they want to do a frontal attack. The last sentence of the paper reads, "We expect the transcriptomic, proteomic, metabolomic, and functional analysis of M4B will uncover pivotal molecule aspects underpinning the regulation of the sophisticated Rhiptortus Burkhardia gut symbiotic association." So they're willing to invest a lot of effort.
Hope they get funded. It's tough.
Yeah.
I can see it might be tough to do that sort of thing. I wonder if is this
well remember that this is an agriculturally important pest.
Okay.
So there's reason for really wanting to know what's going on.
Good. I'm glad, because I think it's really interesting. Do you think, Elio, that this kind of regulation of ectosymbiosis would be more general?
Well, you can argue that we have an M4B region in our stomach.
Mm-hmm.
Our stomach is like the M4B region. It makes sure that, but it's, that's sort of non-specific. The acid of the stomach keeps some of the bacteria from the ingested food from developing. It's more specific. So I don't know. This is there are probably two ways of thinking about it. One is, in fact, non-specific. There are times when you want to keep bugs down in general, regardless of the bug, and there are times when you're dealing with a specific one, and it's just a case-by-case question, I think. Mm-hmm. But it would be surprising if there weren't examples like this all over the place. Sure.
Yeah. Well, especially in other insects.
Easy for me to say. Sure.
Well, you can say that. It's fine.
So, Vincent, is your question whether there are other bacteria that live outside host cells that have an effect on host physiology, or did I miss the question?
I wanted to know if in other cases where we know there are bacteria living outside the cells, are there mechanisms that regulate their abundance as we have here?
Well, look, you could argue that in the large intestine, there isn't such a thing, because the population reaches about saturation. You get 10 to the 12 bugs per gram of feces. So, how many more do you want? It doesn't look like that has been kept down.
But there are clearly examples of overgrowth of individual species, with C. diff being one of the
sure,
right.
Right.
But I think we don't know whether it's whether there are host components involved in suppressing the populations, or whether it's entirely competition with the other bacteria. Because clearly, disruption of the microbiome or the microbial community to C. diff flourishing and becoming a dominant organism.
So it could be that these insects have, I guess they do have a microbiome in addition to this symbiont, right?
They probably do. Imagine in the cecum, they probably have a lot of bugs. Most insects do.
So could those bacteria be producing the inhibitor?
The one in the cecum? It seems unlikely. I mean, it would have to go upstream. That seems unlikely.
in these midgut regions, there are no other bacteria? Not
much.
Usually the midgut does have other bacteria. I don't know this particular insect.
Others do, sure. Others do. But this one doesn't sound like it has a lot. Yeah.
Well, I think it's an interesting hypothesis that the other bacteria are actually producing the compound, and it's being mistaken for a host compound.
Yeah.
Yeah. That's interesting. Yeah. They should look at that for sure.
Well, maybe they'll listen and get an idea. Wouldn't be bad.
That'd be nice. Glad to oblige.
Okay. All right. Thank you for that, Elio.
I have some current event I wanted to to describe. When I was drinking my coffee this morning, thinking about TWiM this afternoon, and reading the New York Times, I was delighted to see microbiology in the editorial section of the New York Times.
Wow.
So not the op-ed, but the editorial section. So they cited a recent paper in Science that did some really neat work looking at the soil microbiome in prairie lands. So this group it was a collaboration from multiple institutions. The senior author is from Kentucky, but they went to, of course, the farmland now out in the prairie that's been cultivated and used for modern agriculture for decades, sampled that, and then compared it to cemetery soils, which had not been disturbed, and found a completely new phylum, or a phylum that is not typical in the tilled soil. So it was just very cool to see microbiology being discussed on the editorial of New York Times.
Can you send me that link? I'd love to put it in the show notes.
Sure will.
That's great. Always nice when microbes...
National Public Radio has been doing a series in the morning on the human microbiome, and they've done a fantastic job of explaining the experiments that have demonstrated the importance of the microbiome. The most recent one I heard was on the impact of the microbiome on mood and behavior, which is a growing area that, depression seems to have a microbiological component, and many other behavioral aggression in mice, other behaviors. And so I think it sort of transforms that whole area of neurobiology and behavioral biology, transforms microbiology and what the breadth of the diseases that we study as microbial diseases.
Wow. That's amazing.
I think the four of us would agree microbiology is a growth area.
Sure is.
Come join us.
Wow, that is great.
We should think about maybe having a podcast dealing with microbial endocrinology, somebody called it,
Yeah.
It's an interesting subject, all right.
Yep.
There's a lot to it. There's a lot of information.
That's amazing. I have a few emails I'd like to read here. The first is from Robin, who I think is writing about some things we have referred to in recent TWiMs. The first one, he writes, liquid water without bacteria on Earth. I don't know when this came up, but he writes 5 sources: endolymph, aqueous humor, urine, cerebrospinal fluid, and amniotic fluid.
All true, but that's not quite what we had in mind. The whole thing started with a Talmudic question on our blog, which asked, are there in fact places other than a sterile broth in a flask and other than inside the body? Yeah, that was the question. So, the intent, obviously this is true. Most of the body, which is not contiguous with the outside, is in fact sterile. But that's not, we know that. I mean, that's obvious. Let's ask a more interesting question. Are there places in nature out which have liquid water and which have no measurable microbial biota? And that, I think, is the answer that I come up with repeatedly is no. But, that's worth asking.
Right. That's the question we ask. That's right. Not on our bodies, right?
Yeah. In our bodies.
And then he continues, ergot on the forehead, close but no cigars. Ergotamine. I guess we mentioned some of these, and he's explaining. Ergotamine is used medicinally for treatment of acute migraine attacks, sometimes in combination with caffeine. Dihydroergotamine is an ergot alkaloid used to treat migraines. Spores blow in. Bacteria, if spore formers, may do likewise. Tinea versicolor, Pityriasis versicolor. Recent research has shown that the majority of Tinea versicolor is caused by the Malassezia globosa fungus, although Malassezia furfur is responsible for a small number of cases, mostly a cosmetic problem. Robin, by the way, is a retired emergency room physician. And finally, microbiotum and microbiogenome are his suggestions for new terms, Elio.
Okay. Let me have it because we conducted a poll in the blog about whether the microbiome refers to the bacteria or the microbes, or does it refer to the genes of the microbes? And I'm going to report on the results of that with a commentary of mine, so I'll be glad to include this.
All right. The next one is from Don. Greetings and the usual well-earned accolades. I have a few questions about the protective effect of melanin in the gut on the reticuloendothelial system. If melanin or its precursors are truly insoluble, how is the bone marrow protected? Were the mice restrained during radiation to shadow the bone marrow by the gut laden with mushrooms, or is a melanin precursor responsible for the remarkable effect?
I think I know the answer to that. It turns out that when you irradiate mice, the overwhelming damage is to the intestinal cells.
Right.
So although the blood cells are affected too, I don't know if it's because there are fewer of them. The gut is a great component of the body, but you want to worry about damage to the intestinal cells. So the melanin doesn't have to do anything other than be there and shield the intestinal cells. At least that's what I read.
So what they said in the paper also is that they believe there are some lymphoid cells in the gut that are protected by the melanin, and those somehow go to the bone marrow and help reconstitute or protect it in some way. So that's that. To have fed refined melanin instead of the Trader Joe's fungi would answer a lot of questions. It is usually the failure of the bone marrow that has killed those people with radiation poisoning, and a protectant would aid such mundane efforts as a manned Mars mission. Thanks again for your gift to science. Well, Don is an MD, so he's curious about this, as many are. We have a letter from Marion who writes about, Elio, do you remember the letter about how you would get proton gradient-driven ATP synthesis in gram-positive bacteria?
Oh, boy.
I don't know if it was last time.
Where is Michael when we need him?
Well, anyway, we couldn't answer it. He said, here's originally Nick's question. I do not understand how this is accomplished, that is ATP synthesis through chemiosmosis, in gram-positive bacteria. And so Marion writes, 2 membranes are not required for proton gradient-driven ATP synthesis. Proton pumps in electron transport chains in the prokaryotic cell membrane concentrate protons inside the cell. This drives membrane-bound ATP synthase, producing ATP inside the cell. In fact, mitochondria are hypothesized to have been derived from endosymbiosis of the ancestors of purple sulfur bacteria. A perquisite of teaching undergraduates.
I have such respect for people who do that. Let me tell you, having spent most of my life in a medical school, I was surrounded by people who knew a great deal about very little. Mm-hmm. And that is not what you do.
I resemble that remark.
What's that?
I said I resemble that remark.
But if you teach in a college setting, you can't afford to have that attitude. You have to really know a great deal about a lot of things.
Yeah, you get questions.
My hats are off for that.
Yeah, you get totally unexpected questions. Yeah, you have to know it. And our last email is from Greg, who writes, Dear TWiM hosts, the last few episodes have been great as usual. I want to comment on 2 things. 1, I feel the term microbiome should refer to the set of microbes living in a given environment. It seems to me that there needs to be a term for this idea in any case. As for the set of genes or genomes present in a given sample, am I wrong in thinking that the word metagenome is adequate for this? Besides, what exactly do we mean?
Oh, so Greg.
Sorry?
I love Greg. Thank you.
The audience may not know that we have on our microphone the author, the originator of the term metagenome. This is Joe's invention.
Yeah.
But even more importantly, I think microbiologists have to be more consistent with the rest of ecology, and biomes are not in the other form of ecology, the macroecology world. Biomes are not genes. Biomes are environments and the collective organisms within them. So I think it's really dangerous for microbiologists to redefine words or suffixes.
Right. Greg continues, besides, what exactly do we mean when we add "ome" to the end of a word? A genome is a set of genes, well, sort of. A proteome is a set of proteins. It seems logical that a microbiome should be a set of microbes. We have the added bonus that a microbiome can also be a biome. I don't know, but perhaps the word was even used in this sense before everyone started overzealously adding "ome" words to our word "ome."
Touché.
Nice. Point number 2. The story from TWiM 67 about melanin-containing fungi living in environments with a lot of ionizing radiation was indeed fascinating, but I am inclined to feel skeptical about the idea that these fungi are actually deriving energy from that radiation. Is there any evidence that the fungi harness the energy of ionizing radiation? I am neither a microbiologist nor a nuclear physicist, but my first guess would be that the fungi are merely unusually resistant to radiation. Perhaps they are very good at repairing damage to their DNA.
Well, that's not quite the basis for this statement. In the paper by Casadeval et al., what they showed is that exposure to radiation enhances the growth of the molds. They're not just resistant, they grow better in the presence of radiation. And if you make an albino mutant, one that doesn't have melanin, you do not have that effect. They are just the same as the controls. So the statement here is not just resistance, but enhancement of growth.
Okay.
And I find personally that this is not just mysterious, but incredibly exciting, because it would it's a paradigm shift in a sense of what we know about the utilization of electromagnetic waves for energy purposes. I mean, we only have one good example, and that's photosynthesis. And here we have the possibility for something that maybe doesn't take place on the same scale. Maybe it's not as important from a practical point of view, but it sounds like this is an alternative, a new thing. Oh. I like it.
Okay, I'll finish Greg's point here. This resistance would allow them to thrive where other organisms can't, even while using conventional sources of food. Although I can't say I understand photosynthesis, my impression is that there is a good understanding of how the energy of visible light can be harvested by chemical means. The energy of visible photons must be a good match to the energy of a useful transition between states of a molecule. Of course, ionizing radiation also couples to chemical changes since it can blow molecules apart. But is there any way to capture the energy when this happens? I haven't heard of anything like this. It is worth finding out, but as Carl Sagan said, extraordinary claims require extraordinary evidence. I am a great fan of TWiV, TWiP, and TWiM. Keep up the great work, Greg. Is there any way to capture this ionizing radiation? Do we know this? I don't think so, right?
Well, melanin certainly is altered by ionizing radiation.
Yeah.
It is if there is an antenna, equivalent of a chlorophyll antenna, then melanin is a good one. So, it's plausible. That's about it. The data are very scarce. The experiment has been repeated, but there is not a big literature on it, and I'm surprised. I would think that this deserves a lot more attention.
Yeah.
And Arthur Casadeval thinks so too. He agrees.
I've sent him this email, so we'll see next time. Maybe he'll have a response.
That'd be nice.
All right, that's TWiM 69. You'll find this episode and all the others at iTunes and also at microbeworld.org/TWiM. And if you have questions or comments for us, as you hear, we love reading them, send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thanks for joining us today, Michele.
My pleasure.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Again, my pleasure too.
And Joe Handelsman is at Yale University. Thanks for joining us today, Joe.
Oh, thank you so much for having me. If I'm not on for a while, it won't be because I don't love TWiM.
Always happy to hear you, and so whatever you can do would be great.
We hope you come back soon.
I'm Vincent Racaniello, and you can find me at virology.ws. I would like to thank the American Society for Microbiology for supporting TWiM, and Chris Condayan and Ray Ortega for their technical help. The music that you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.